Welcome to another video where we continue answering biology questions. So we're going to answer questions based on molecular biology and genetics. So here are the questions. Question 1. Which of the following is not a property of water? So as usual here we have 6 options. So option 1 up to 5. You can choose from any of these. Then option 6 is I don't know. That is, if you're not sure the answer to a particular question, you go for this option. So if you put option 6, you're going to get a 0. And if you put any of the option 1 up to 5 and you tend to get that question wrong, so you get a -1. Yeah, so that's why we have option 6 there. It's there to save you from negatives. So which of the following is not a property of water? 1. It has a high surface tension. So this is true. It has a very high surface tension. That is the reason why some insects are able to walk on water. That is the surface tension of water. So it has high surface tension, it's true. 2. It has a high heat capacity. So this is true. It takes a lot of energy for you to change the temperature of water. 3. It requires a lot of heat energy for it to be converted from liquid to gas. So we know that the boiling point of water is 100 degrees Celsius. So you can imagine the energy that you need for you to reach 100 degrees Celsius. So it's true that it requires a lot of heat energy for you to change the temperature. Then we have option 4, which is saying it is a non-polar solvent. So water is a polar solvent and not a non-polar. Non-polar has to deal with equal distribution of electrons. So we have seen, or we know that water is made up of hydrogen and oxygen. So there is an equal distribution of electrons in a water molecule. Therefore, water is a what? Is a polar solvent and not a non-polar. A non-polar molecule is something which has got equal distribution of electrons. A good example can be chlorine or chloride ion. So this is a non-polar molecule. So water is a polar molecule because there is an equal distribution of electrons, where we see each hydrogen atom giving an electron and oxygen is giving out 2 electrons. So this is a non-polar property of water. So option 4 seems to be a false statement. 5 is saying water is a dipolar. Dipolar, that is, it has got 2 charges, which is true. So we know that we have got delta negative and delta positive. So these are what? The dipolar nature of water, that it has got both negative and positive, though these are partial charges. So the answer to question 1 is 4. 4 is the first statement. Water is a polar solvent and not a non-polar. Question 2. Nucleotides are composed of. So what are nucleotides, first of all? So nucleotides, these are what? These are monomers of nucleic acids. So nucleic acid, here we are talking about DNA and RNA. The monomers of these molecules are what we call nucleotides. So what makes up this nucleotide? So each nucleotide molecule is made up of 3 components. And these 3 components that makes up a nucleotide molecule is that we have got a nitrogenous base, then you have got a pentose sugar, not just another sugar, but a pentose sugar, plus a phosphate group. So these are the 3 molecules that makes up the monomers of nucleic acids, which are DNA and RNA. So nucleotides are ribose and proteins, no. Deoxyribose and phosphate, no. So sugar, nitrogenous base, and the protein, no. So you have to be very careful here. Make sure that nucleotide, you have got these 3: nitrogenous base, a pentose sugar, and a phosphate group. Nothing else in replacement to any of these 3. Then 4 is saying nucleoside and the phosphate. Then 5 is saying nucleoside and the sugar. So if you check the options here, we don't have an option which has got these 3: a base, a nitrogenous base, sugar, and a phosphate group. So you should also be familiar with what a nucleoside is. So a nucleoside, this is a molecule which is only made up of two things, a nitrogenous base and a pentose sugar. That molecule is what you call a nucleoside. A nucleoside and a nucleotide, these two are not the same. A tide is a monomer of nucleic acid. A nucleoside, this is just a molecule which is made up of two things, a nitrogenous base and a pentose sugar. So if you add a phosphate group to a nucleoside, then what you have now, you have what? A nucleotide. So option 4 is the correct statement here. A nucleoside and a phosphate, that is what? A nucleotide. Yeah, so the answer here was option 4 for question 2. Then we'll move on to question 3. So question 3 here reads, which of the following structures is found in both prokaryotic and eukaryotic cells? So which of these structures is found in both? So we're looking for that organelle which is present in both prokaryotic and eukaryotic cell. So one is saying Golgi apparatus. So Golgi apparatus is not found in prokaryotic cells. Endoplasmic reticulum is not found in prokaryotic cells. Then mitochondria. Mitochondria also is not found in prokaryotic cells. Mitochondria is only found in eukaryotic cells. The nucleus. So prokaryotic cells do not have a nucleus. Instead, they have what we call a nucleoid. A nucleoid, this is a structure that keeps up the genetic material, that is DNA. So DNA in prokaryotic cells is stored in an organelle known as a nucleotide and not a nucleus. Then 5 is saying ribosomes. So ribosomes, these are required for protein synthesis, and we do know to say that both prokaryotic and eukaryotic cell, they have to synthesize protein. So they both need what? Ribosomes. Therefore, ribosomes from the given option is the only organelle which is found both in prokaryotic and eukaryotic cell. Question 4. Proteins are modified in the dash or the eukaryotic cell. So as we said earlier, that some organelles are not found in prokaryotic cell. So eukaryotic cells have got a special organelle which modifies proteins. After the end of protein synthesis, you are going to have what? A primary protein, primary structure of protein, which is just a chain of amino acids. So this has now to go, a polypeptide chain has now to go to an organelle where it has to be modified, either to be a secondary protein, a quaternary, or a tertiary structure. So that modification is done by a special organelle. So that's what this question is asking us, like which of these organelles given here is responsible for modifying proteins in eukaryotic cell. So smooth endoplasmic reticulum, no. Golgi apparatus. So this is an organelle which is responsible for protein modification. Ribosomes are just there for protein synthesis, nucleus, and nucleolus. Nucleolus, this is an organelle which is found within the nucleus, and this is the organelle which keeps the DNA in the nucleus. So the organelle that is responsible for modifying proteins is Golgi apparatus. So some books will say Golgi bodies. It's one and the same. Question 5. Why is the amino acid in proteins polar? So first of all, we have to understand what polar is. So polar, we said, this is an equal distribution of electrons. An equal distribution of electrons. That is, if you have two atoms, okay, making a compound. Let's say if you have this. So here you have got nitrogen and oxygen. So if these two atoms which are involved in the formation of this compound, the electrons which they share amongst themselves is not the same. That molecule is what we call a polar molecule. There is an equal distribution of electrons. Let's say if this is giving out 3 electrons and this is only giving out 2 electrons, this is what is what we call the unequal distribution of electrons, and this is what makes a compound to be polar. If this nitrogen is giving out 2 electrons and oxygen is also giving out 2 electrons for this bond to be formed, then this is what? There is equal distribution of electron. Therefore, this molecule is going to be termed as a polar is going to be termed as a non-polar molecule. A good example of a non-polar molecule can be cooking oil. In cooking oil, there's equal distribution of electrons, so it's non-polar. Even a chloride molecule. Okay, so what makes amino acids in proteins polar? What makes the unequal distribution of electrons? So you have to know that the structure of amino acid, you have got this group, the amine group, then you have got carbon, then here you have this carboxyl group, then here you have got the side chain and a hydrogen. This is the general structure of amino acid. So what makes this structure polar? One is saying electrons are shared equally between nitrogen and hydrogen atoms. So as long as there is equal distribution, then that particular molecule cannot be polar. It's non-polar. Two is saying nitrogen is more electronegative than hydrogen. That is, electrons spend more time close to the nucleus of nitrogen than to the nucleus of hydrogen. Or come. Then three is saying nitrogen forms ionic bonds with a hydrogen. So, and the type of bond that is formed between nitrogen and hydrogen is covalent bond. Okay, they share electrons. It's not an ionic bond. There's no sharing. There's no an atom which loses the electrons to the other atom. Then four is saying nitrogen has acidic properties. Nitrogen has acidic properties. So, nitrogen is a base. So meaning that it has got basic properties. It doesn't have acidic properties. 5, nitrogen is more electropositive than hydrogen. So electropositive, that is, it's more, it can give electrons more than what? Nitrogen. Oh, not more than hydrogen, which is not true. So nitrogen is more electronegative than hydrogen. That is, electrons tend to spend more time close to the nucleus of nitrogen in this amine group. Nitrogen is more electronegative than hydrogen. On the periodic table, we said that the electronegativity increases as you move from left to right. So on the periodic table, you are going to find that nitrogen comes first before I think before oxygen. So this nitrogen is more electronegative. We have hydrogen somewhere. I think we have something like this on the periodic table. So as you move from left to right, the electronegativity tend to increase. Okay, I think if I've not mistaken. Yeah, so nitrogen is more electronegative than hydrogen. So that's the reason why the amino group in proteins is polar. So there is an equal distribution of electrons. That is because these electrons in hydrogen, they will be more close to the nucleus of nitrogen. As a result, this hydrogen is going to attain a partial positive charge. Then this one is going to attain a partial negative charge. Therefore, it will make that group, the amine group, to be polar. 6. Identify the incorrect statement about enzymes. 1. They are much larger than their substrate. So this is true. Okay? So enzymes are much larger as compared to their substrate. Then 2 is saying only a small portion of their structure is directly involved in catalysis. That is the active site. So you are going to have an enzyme. Let's say this is an enzyme. Then you have got that is the active site. So it is on the active site that is involved in the catalysis. That's why it's saying on the small portion of their structure is directly involved in the catalysis. So it is on the active site, which is the small portion of this entire enzyme. 3 is saying the lock and key model explains the enzyme specificity. That is true. So the lock and key theory, this is based on the active site and the substrate which is going to catalyze this enzyme. So it has to be specific. That is, the substrate that is going to catalyze this enzyme should also have this structure. Okay? Should have this structure of the active site. So which is true. 4 is saying enzymes work by raising activation energy needed for the reaction. So 4 here sounds to be false statement. 5 is saying different enzymes have different amino acid arrangements in their active site. So the amino acid arrangement, this is based on the side chain. The general structure that I gave you previously, we said that each amino acid has got this group, then the R group. Each amino acid has got a different R group. So this R group is what determines the active site of this enzyme. So different enzymes have different amino acid arrangements in their active site because different amino acids have got different R groups. So it means that even the active site is going to have what? Different arrangement in their active site. So the first statement here is option 4. Enzymes work by raising the activation energy needed for the reaction. Initially, enzymes reduce the activation energy. So let's say if the initial energy to complete this reaction was supposed to be like this, okay? Then let's say this is 70. When you introduce an enzyme is going to reduce this activation energy. It will provide an alternative route that is going to produce results more faster than it was supposed to. So it's going to lower this activation energy. Let's say maybe just to there. So let's say this is maybe 30. So from 70 to 30. So it decreases, it does not raise. It does not rise the activation energy. Instead, it reduces. So 4 is the fourth statement with respect to enzyme. So hence, that's the incorrect statement for this question. Question 6. 7. Identify the structure that is not found in prokaryotic cells. So we know that some organelles miss in prokaryotic cells. Plasmid, yes, these are DNA which can be transferred from one bacteria to another. So they have plasmid. Cell membrane. All living organisms have got cell membrane. Mitochondria. So earlier on we said that prokaryotic cells do not have mitochondria. Okay? So this is not found in a prokaryotic cell. Ribosomes, they all have cell wall. So prokaryotic cells do have cell wall. They have got a capsule and a cell wall, which prevents them from phagocytosis. So the only first statement here, or the structure that is not found in prokaryotic cell, is a mitochondria. Hence, the answer to this question is option 3. 8. Which of the following is not true about transfer RNA? So which of the following is not true about transfer RNA? A is saying it has got a D arm, true, T arm and codon arm, amino acid acceptor arm, yes. The CCA sequence is found at its 5' end. So the structure of tRNA is like this, okay. So it's like this. Then this is the 5' end. This is the 3' end. This is the structure which has got this, the CCA sequence. The CC sequence is at the 3' end, which we call the acceptor arm. That is where the amino acid is attached, the acceptor arm. So it has got the D arm. Yes, this is the D arm, the T arm, then this is the anticodon arm, and also the amino acid acceptor arm, which is the 3' end. So, which of the following is not true about transfer RNA? So the first statement here is option 5. Option 5 is saying the CC sequence is found at the 5' end. This is false because the CC sequence in tRNA is found at the 3' end. In short, it is found at the acceptor arm, which is the 3' end. So the first statement here, or the statement which is not true, is option 5. Hence, that's the answer. Okay, that's the answer to this question. Then question 9. Select the statement that is true about plasma membranes. So 1 is saying all living cells are surrounded by plasma membrane, which is true because in the previous question, which was I think question 7, we had this similar question about plasma membrane. So I said all living organisms have plasma membrane, be it prokaryotic or eukaryotic cell. So 1 is true. Cells receive chemical messages from the environment through the plasma membrane. That is true. So these chemical messages, they have to pass through what? The plasma membrane. Okay. Then 3 is saying the plasma membrane have protein channels through which nutrients for the cell can pass. That is true. So we know that it has got the integral protein and transverse proteins. Okay, those provide channels uh, through which nutrients for the cell can pass. Cholesterol is part of the plasma membrane. This is a true statement. Then 5 is saying all the above statements are true. So you see that option 5 combines all of them. And for the fact that from option 1 to 4, we have found that all of them are true about plasma membrane. So the correct statement or the correct answer to this question is going to be option 5. All the statements above are true about plasma membrane. So that's the answer to that to question 9. 10. A prokaryotic cell lacks. So we have discussed much more about prokaryotic cells. So by now we have to be finding it very easy to answer this question. A prokaryotic cell lacks plasma membrane. So we say all living organisms have plasma membrane, nuclear envelope. So this is true. Plasma membrane do not have nuclear envelope. Why? It's because nuclear envelope, this is a structure which surrounds the nucleus. So since we say that prokaryotic cell lacks nucleus, meaning that they don't have what? Plasma membrane or they don't have nuclear envelope. Okay? So they don't have nuclear envelope. They have cilia, these are projection on the cell surface. Then ribosomes, they do have flagella, this is what they use for locomotion, so they do have. So they only lack nuclear envelope because they do not have the nucleus, because nuclear envelope is a structure which surrounds the nucleus. So the answer there is nuclear envelope. 11. Which of the following is true about enzyme cofactors? So what are enzyme cofactors? So cofactors, these are non-protein structures which helps in catalysis. So if you have got an enzyme, if you have got a protein, then this protein is attached to, let's say, maybe a metal ion. So like a good example can be hemoglobin. We know that in hemoglobin contains iron. So that ion is a cofactor. So it is a non-protein portion of an enzyme which helps in catalysis. That is what a cofactor is. So a cofactor can be a metal or an inorganic compound, even a vitamin. It can also be, as long as that particular molecule is a non-protein component, but it is part of the protein. That is a cofactor. So it's there just to help in catalysis. So one is saying cofactors are always proteins, so they are non-proteins. 2 is saying enzymes are found without the presence of cofactors. So some enzyme, a good example is, but when we say enzymes and protein the same because enzymes are protein in nature. So enzymes can function without the presence of cofactor. So a good example is hemoglobin. Hemoglobin has got a cofactor. So some enzymes cannot function without a cofactor. Then 3 is saying cofactor is primarily involved in the stabilizing the enzyme's tertiary structure. So not really, because even some primary structure, quaternary structure protein, they have what? Cofactors. Then 4 is saying cofactors are essential for enzyme catalytic activity. So this is much close. 5 is saying cofactors are carbohydrate in nature. No, it can be a metal, even an inorganic compound. So option 4 here is going to be the correct statement. Cofactors are essential for enzyme catalytic activity because some proteins cannot function without these cofactors. So they need cofactors for them to carry out their function. Therefore, the answer there is option 4. Question 12. Question 12 reads, bacterial cell wall is made up of 1. Cellulose. So cellulose, one thing about cellulose, one thing that we know about cellulose is that it is involved in a structural component. Okay, is involved in a structural component. So bacterial cell wall is made up of cellulose, no, polysaccharide. These are what? These are these are carbohydrates. 3 is saying it's made up of peptidoglycan. 4 is saying hemicellulose. 5 is saying wax. So the bacterial cell wall is made up of peptidoglycan layer, and this peptidoglycan layer is the one which plays an important role when it comes to gram staining. Okay, so some gram-positive bacteria, they have got a thick peptidoglycan layer. Some gram-negative have got a thin peptidoglycan layer, but the base is that all bacterial cells are made up of peptidoglycan. So that's the answer there. Then 13. The number of dash is equivalent to the atomic number. So what is an atomic number of an atom? So an atomic number of an atom, if I give you, let's say, sodium. Sodium has got a mass number of 23. The number that you see down there, that is the atomic number. So the atomic number of sodium is 11. Okay? So it's saying that the atomic number, the number of dash is equivalent to the atomic number. This atomic number, we say that is equal to the number of electrons of an atom. If a particular atom has got an atomic number of 12, it means that it has got 12 electrons as well. If it has got 17 as its atomic number, it means that it also has got 17 number of electrons. So the number of electron is equivalent to the atomic number. Okay, so this atomic number is also determined by the number of protons. Okay, the number of protons. So these protons are found in the nucleus. The nucleus of an atom contains neutral particles, which are known as neutrons, and also positive charged particles, which are known as what? Protons. Okay, so these protons are what determines, or what gives out the proton number of an atom, or atomic number of an atom. So atomic number is equivalent to number of protons of an atom. The number of protons is equivalent to number of electrons of an atom. So the number of protons is equivalent to the atomic number of an atom. So option 5 for question 13. That's it. Then I'll move on to 14. So 14 reads, which of the following is a normal pair of nitrogenous base in DNA? So in DNA, you know that you only got 2 pairs of these nitrogenous bases. In short, the nitrogenous bases that you find in DNA are that you have got adenine, you have got cytosine, you have got guanine, and then you have got thymine. These are the only 4 nitrogenous bases which you find in DNA. We don't have uracil in DNA. So the question is asking about the normal pairing of nitrogenous bases in DNA. So in DNA, adenine will always pair with thymine through a double hydrogen bond, whereas guanine will always pair with cytosine through a triple hydrogen bond. This is the normal pairing of nitrogenous bases in DNA. So guanine, cytosine, true. Uracil, adenine, we don't have uracil in DNA. Cytosine, adenine. Cytosine will always pair with guanine. Then thymine, uracil, we don't have this pair. Thymine and cytosine, we also don't have that complementary base pairing. So the answer is option 1. Okay? Guanine will always pair with cytosine, be it in DNA or RNA. That pairing does not change. In the electron configuration of the oxygen atom, how many electrons occupy the second energy level? So the second energy level. So we know that we have 1s, then we are going to have 2s and 2p. I think we can end here. So the s orbital can only occupy 2 electrons. This will also occupy 2. Then this has to occupy a maximum of 6. So we know that oxygen has got 8 electrons. So 2 plus 2, 4. So we are only remaining with 4. So in the energy level of this atom, the first energy level, this is the 1s. So this only occupies 2. Then you have got the 2s. The 2s has got occupies 2 as well. Then we have got the 2p. The p orbital has got 3 orbitals like that. So since we have 4 there, we're going to have this, that, and this, then this. So 4 there. So the question is, in the electronic configuration for the oxygen atom, how many electrons occupy the second energy level? So the second energy level is this one here, or the second shell in short. So how many electrons occupy the second energy level? So the second energy level is occupied by how many electrons? 2 plus 4, so 6. So the answer there has to be option 3. So 6 electrons occupy the second energy level. The second energy level is the second shell of an atom. So you combine both the S and the P atoms. Electrons. 16. Which of the following is not a function of bacterial cell? So bacterial cell has come back. So we said earlier on that the function of bacterial cell is that one, it gives the cell shape. It gives shape to the cell. Then also it protects the cell from phagocytosis and also from bursting. Okay, all those. So gives shape and rigidity to the cell, which is true. Protects the cell from rupturing, which is also true. Then surrounds the plasma membrane. So the cell wall surrounds the plasma membrane because it is the outermost structure of the bacteria. So you're going to have a cell wall outside, then you have the plasma membrane inside. So this plasma membrane is surrounded by the cell wall, which is true. It is the site for the of action for some antibiotics. So antibiotics, these are drugs. When you take some of these drugs, example is penicillin. So penicillin is going to interfere with the cell wall. The thing will be to degrade this cell wall. When this cell wall is degraded, this bacteria cannot survive, it will die, and that is how some antibiotics works. Okay, so it is true. Then 5 is saying contains genetic material. So the genetic material in bacteria cell, they are not found in the cell wall. They are found in the plasmid. If not in the plasmid, then they are found in the nucleoid. These are the two organelles in the prokaryotic cell which contain genetic information or genetic material. So 5 is the first statement with regard to cell wall. Then 16. Okay, so there's a missing page. We jump to 22. 22 reads, in an atom nucleus, the attraction of electrons to protons is by. So in an atom nucleus, so you are going to have, this is an atom, then you have got a nucleus. The attraction of electrons to protons is by. So you know that in the nucleus you have neutrons and protons. Protons are positively charged. Then in the shells, this is where you have electrons. Okay, electrons are negatively charged. So the attraction of electrons to protons, these electrons are always attracted to the protons because these two have got different charges. So this attraction of electrons to protons is by the effect of the outermost shells. 2, the effect of the p orbitals only. No, so some atoms do not even have p orbitals. A good example is hydrogen. So we can't say the effect of the p orbitals only. Electromagnetic force, 4 nuclear force, then 5 is saying neutrons. Okay, so the attraction of electrons to protons is by the electromagnetic force. So electromagnetic force, this is the force of attraction which exists between positively charged protons in the nucleus and negatively charged electrons in the shells of an atom. So that force of attraction is what is known as electromagnetic force. So this is the force that makes these electrons to be attracted to the protons which are in the nucleus of an atom. Okay, nuclear force, this is just the force within the nucleus. So you have got neutrons and protons. So the force that protons possesses, that is the nuclear force. Okay, 23. Cellulose is non-reducing sugar because of the presence of the dash glycosidic bond. So first of all, you have to identify the monomers of cellulose. So the monomers of cellulose are, you have glucose, you have glucose, and alpha glucose for that matter, and fructose. Okay, this will make cellulose. Okay, so this bond is between carbon 4 of glucose and carbon 2 of fructose. So this is the alpha glycosidic bond, because you have got alpha glucose and you have alpha glucose and a fructose molecule coming together to form this bond. And for this bond to be possible, this fructose has to turn 180 degrees for this bond to be formed, because you know that fructose takes up the I think it's the furanose ring, has got a furanose ring, okay, something like this. It doesn't look like glucose, which looks like this. No, it takes up this structure. So the carbon 2 is at this oxygen. So for this bond to be formed, or this carbon, for this bond to be formed, because carbon 2 of glucose is here, for that bond to be formed, this has to turn 180 degrees so that it becomes something like this. Okay, then now the bond will take place between carbon 2 of fructose and carbon 4 of glucose. So this bond is going to be alpha glycosidic bond, because the glucose involved there is an alpha glucose. So the bond is going to be this is carbon 2, not carbon 4, sorry. The carbon 2 of glucose, carbon 4 is going to be here. Okay, so it's between carbon 2 of glucose and what am I even? No. So this has to be carbon 1. Carbon 1, because glucose is like this. I've confused you a lot. Okay, so glucose is like this. So this is carbon 1. This carbon 1, this is where you have got this. So alpha, that is above. If the hydroxyl group at carbon 1 is above, then that type of glucose is what? Is alpha. If it's below, then it's so above is beta. Above is the opposite. Above is beta glucose. If it's below starts with B, then it's alpha. Okay, yeah. So this is how I remember them. So if it's above, then it's a beta glucose. If the hydroxyl group at carbon 1 of glucose is below, then it's what? It's alpha. So this type of carbon, this type of glucose which is involved in the bond formation between glucose and fructose to form a sucrose, it's an alpha glucose. Therefore, it's going to be 1 to 2 alpha glycosidic bond. The 1 is for the carbon 1 of glucose, and the 2 is for the carbon 2 of fructose. That is how the bond is formed in a sucrose molecule, where fructose has to turn 180 degrees. Then 24. Which of the following is not a function of carbohydrates in living organisms? 1. Energy storage. That is true. A good example is glycogen. Glycogen stores energy. 2. Instant energy. So the stored energy can be converted at any time. That is instant energy. When you are undergoing fasting, the energy that will be being used is the energy that was stored. 3. Production of roughage in the human digestive system. This is true. Structural support. A good example is cellulose, which is true. Genetic information. So carbohydrate does not store genetic information. So this is the fourth statement. Therefore, it's the correct answer for this question, because we are looking for what is not the function of carbohydrate. So genetic information is not the function of carbohydrate. 25. Water is able to expand when it freezes because of the formation of covalent bonds. So what happens when you freeze water? So when you freeze water, it forms what we call lattice. So lattice is going to create some spaces within a water molecule. So those spaces are what makes water to be less, what makes ice to be less dense as compared to the fresh water, because there are those spaces. And it's also the reason why the volume of water expands when you freeze it, because or those lattice spaces. So water is able to expand when it freezes because of the formation of covalent bonds. So disruption of hydrogen bond. Hydrogen bonds are only disrupted when you are boiling water. There is breaking and joining. Then breaking of ionic bonds. We don't have ionic bonds in water. Increase in density. So when you freeze water, the density reduces because you are creating those spaces, making into water to be, or ice to be less dense as compared to the normal water. The formation of rigid hydrogen bonds. So the water is able to expand when it freezes because of the formation of rigid hydrogen bonds. So these rigid hydrogen bonds are the ones which are going to result in the formation of lattice. How is it? So it's because we know that a water molecule, each water molecule can be linked to a maximum of 4. Okay? To a maximum of 4. So here, there, the other one here, and that one there. Then this particular water molecule is 4. Then another water molecule the same. So there are going to be those rigid hydrogen bonds which are going to be formed, thereby resulting to water expanding in size and creating those spaces. So the answer there is option 5. 26. The molecule below is so if you look at this molecule, is a polysaccharide because it's a carbohydrate. So it's a polysaccharide, but what type of molecule is it? What type of a polysaccharide is it? So we have to check the monomers which are involved in here. Okay. So if we look at this molecule, we have got oxygen here. For this one, the oxygen is there. This one is here. That one is there. So in short, the incoming monomer changes its direction. This one is upside down. Okay. This one is upside down. Then this one is normal. This one is also upside down. This one is normal, meaning that the incoming should also be what? Upside down. Okay. So what monomers are these? So we have to check carbon. We have to check carbon 4. Okay. So this is carbon 1, carbon 2, carbon 3, carbon 4 is here. So we see the hydroxyl group at carbon 4 is above. So if it's above, then that is what? That is glucose. Okay, that is glucose. Then here, this was okay. This was I think this was above. So above, this one was will be beta. Then will be beta glucose because it's above. Then this one will also be beta glucose. So polysaccharide is made up of beta glucose molecule. So beta glucose molecule in a way that the incoming monomer has to change upside down. So that is cellulose. Cellulose, you know that humans do not have enzymes which can digest cellulose. Why is it so? It's because of this bond formation which is formed here. This bond formation that is formed there is very rigid, such that only animals can digest cellulose. And that is why animals are able to feed on grass, because they have got enzymes which are able to break down cellulose. So cellulose is going to look like this. Okay, you are going to have this, then this hydrogen atom will join these together. So this bond which exists in between these is very stubborn. That is why when you feed on cellulose, the structures of cellulose are going to come out the way they are. They will not be digested because we lack that enzyme. So the answer to this question, the molecule below is it has to be cellulose beta cellulose, which is formed through a 1 to 4 glycoside linkage of glucose molecule. So this is carbon 1, this is carbon 4. That's why the bond here is 1 to 4. The type of monomers which are involved are beta. That's why we are saying beta 1 to 4 glycosidic beta 1 to 4 glycosidic bond. So the answer has to be starch with 1 to 4, no. Amylose with alpha glucose units. 4 is saying glycogen with that. Amylopectin, cellulose with beta 1 to 4 linkages of glucose molecule. So this is the answer here. So for you to get this question correct, we are supposed to analyze the monomers involved and also the monomers involved and also the type of bond. That's how you answer this question. Yeah.